There is a biological switch that tells your muscles when to stop growing. Remove that switch in certain animals and they develop extraordinary muscularity. Some cattle become so muscular that the phenotype is literally called double muscling. A rare child born with disruption of the same pathway develops unusually large and powerful muscles. I've seen it on the internet. The gene involved is myostatin. And now biohackers are asking a dangerous but scientifically fascinating question. What happens if we deliberately suppress that pathway in a healthy adult? Not with testosterone, not with like conventional bodybuilding drugs, but with gene therapy designed to keep producing follistatin, a natural protein that blocks myostatin and related growth regulators. One injection, potentially years of biological activity. More muscle, less fat, perhaps better function with aging. That's the promise. Let's get to it. Here's the problem though. When you're taking an experimental drug, you can usually stop taking it, right? When you alter long-term gene expression, there may be no simple switch to turn it off, even in next generations. Today, I'm going to steel man the case for follistatin gene therapy. Then, I'm going to dismantle the hype. We will examine myostatin, follistatin, Bimagrumab, which I kind of mentioned in a previous episode, muscle mass versus muscle function, and why an unregulated gene therapy injection is not simply an advanced supplement. The science is real. The certainty is not. As former Minister of Health, I've had to evaluate medical innovation from two directions at the same time. So, the first question I ask myself is, could this technology change lives? The second question is, what happens when enthusiasm moves faster than evidence, manufacturing standards, and patient protection? Gene therapy demands both questions. And trust me, I'm not anti, innovation, but in the bodybuilding world, man, and sports world, they will jump on this before the evidence is out. So I'm strongly in favor of responsible human experimentation. But when a treatment may produce long-lasting biological changes, the threshold for evidence should be higher, not lower. So let us begin with the strongest possible version of the argument. So, one, muscle is not a cosmetic tissue. We've been through this, and I'll go through this again. It's a metabolic organ. It stores glucose. It's your glucose sink. It supports insulin sensitivity. It protects your bones. It keeps your bones strong. It allows us to recover from illness. It reduces frailty when we get older. And it's one of the strongest physical reserves we carry into old age. Loss of muscle mass and strength, particularly when it combines with poor balance and low cardiorespiratory fitness, is associated with falls, disability, loss of independence, and mortality. So it's super important. Resistance training and adequate protein remain the foundations, and I talk about this ad nauseum. But many people cannot build or preserve enough muscle through lifestyle alone. Consider diseases such as MD, muscular dystrophy, inclusion body myocytes, severe frailty, cancer-associated wasting, age-related sarcopenia, prolonged immobilization. See how that causes atrophy? For those people, a therapy that meaningfully increases functional muscle could be transformative. Functional muscle. Myostatin is one of the most biologically logical targets we have. Humans and animals with reduced myostatin signaling can develop substantially greater muscle mass. Follistatin naturally antagonizes myostatin and other members of the trend-forming growth factor beta family. And early human gene therapy studies in serious muscle diseases have provided proof that follistatin gene delivery is biologically possible. That is the strongest case. The target is real. The mechanism is plausible. The unmet need is enormous. And muscle preservation could affect not only mobility, but metabolic health and healthy aging. So yes, 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 and yes. It deserves serious research. So what is myostatin? Myostatin is a protein produced primarily by skeletal muscle. It's a gene, right? The gene is called MSTN. Its job is to restrain muscle growth, so it stops the muscle growth. Think of it as the brakes. The body has a braking system. It signals through receptors that activate intercellular pathways, limiting muscle cell growth and protein synthesis. This makes biological sense. We can't just keep growing. The human body is not designed to maximize muscle at any cost, right? Muscle requires energy. It requires blood supply. It changes mechanical loading on the joints and tendons. So, yeah, some is good, too much, no good. Evolution always favors balance, not the largest possible biceps. So when the MSTN gene is severely disrupted, that brake is weakened, the braking system. Certain cattle breeds with myostatin mutations display dramatic muscularity. Whippets with specific variants can become unusually powerful and big. And a rare human case involving loss of functional myostatin demonstrated marked muscle hypertrophy from early childhood. But genetic reality is not the same as treatment trial. Someone born with an altered pathway develops within that biology. That does not tell us exactly what happens when the pathway is suppressed suddenly in a 55-year-old adult with cardiovascular disease, tendon degeneration, or occult cancer. So that's where we need to go. That's where the studies need to go. So what is follistatin? Follistatin is a natural occurring protein that binds several signaling molecules. One of those molecules is myostatin. But follistatin is not a perfect selective myostatin inhibitor. It can also interact with activins and other related protein. That distinction is crucial. So it's not just surgical. It acts with, other proteins in the body. Online discussion often reduce the pathway to more follistatin equals less myostatin equals more muscle. Biology is not linear, as I explained. It affects the whole body. It affects different structures, different areas that we're not looking at. Activin signaling is involved in reproductive biology, inflammation, fibrosis, metabolism, tissue repair, and multiple organ systems. The broader the biological target, the greater the possibility of effects outside skeletal muscle. Different follistatin isoforms also behave differently. FS288 binds more strongly to cell surfaces. FS315 is more freely circulating. FS344 refers to the genetic sequence commonly used to produce the circulating FS315 isoform. So when a clinic advertises follistatin therapy, there's a lot of questions. You know, the first question has to be, what exactly is being delivered? A recombinant protein? A peptide falsely marketed under the follistatin name? A plasmid? I mean, these questions will keep going. Why? Because we need to know. They all have different effects on different areas. Is it a viral vector? Which gene sequence? Which promoter? We're not talking Don King, right? Which tissues will express it? Where is it going to be effect? For how long? How long will the effect last? And how was the material manufactured? So there's a ton of questions that need to be answered. And, you know, they're not technical distractions. They are the treatment. So we have to distinguish what gene therapy actually means. Traditional medication supplies a molecule to the body. Gene therapy supplies biological instructions. In some follistatin studies, an adeno-associated viral vector, or AAV, has been used to carry the follistatin gene into the skeletal muscle. The virus is modified so it cannot reproduce like a normal virus. Its job is to deliver genetic material. The treated muscle cells can then produce follistatin for a prolonged period. That is what makes the gene therapy powerful. It is also what makes it difficult to reverse. With a tablet, adverse effects may diminish after the drug clears, so you can stop, right? And then wait till it clears your system. With durable gene expression, simply deciding that you no longer want the effect may not stop production. The actual duration can vary. Expression may decline. There's so many things that can happen that once we administer it, we have very little control over. Immune response, person to person, may alter it. But you should never approach gene therapy as though it were, a temporary vitamin infusion or a medication that we're used to. Gene therapy is a different platform. We need to think very differently about it. Follistatin gene therapy has been studied in a small number of patients with serious neuromuscular disease, including Becker's muscular dystrophy and sporadic inclusion body myocytes. These studies were early stage, small, and designed primarily to explore safety, biological, and functioning signals. They are not large longevity trials in healthy adults. They did not establish that follistatin gene therapy extends life. They did not prove that it prevents frailty in the general population. And they did not validate every formulation now being promoted by commercial biohacking clinics. That distinction must be made clearly. Evidence that a carefully manufactured vector was studied under one protocol in a small group of patients with muscular diseases does not automatically validate an independent, manufacturer, plasmid, or injection sold to healthy customers in other jurisdictions. Follistatin gene therapy is a category, not one standardized product. Muscle mass is not muscle function. This is the next thing. This is where the story becomes especially important. Multiple drugs targeting myostatin and activin pathways have increased lean mass without producing equivalent improvements in strength or physical function. This is a warning. A larger muscle on a scan is not automatically a more useful muscle. Muscle quality depends on neuromuscular coordination, mitochondrial capacity, blood flow, tendon strength. I mean, there's a ton of stuff, right? Tendon integrity. There's a ton of stuff that really determines whether this muscle becomes useful. Is there motor unit recruitment? Is there intramuscular fat? Is that what blew it up? Training history and the ability of the person to translate tissue into movement. If a therapy increases muscle volume by 10% but does not improve walking speed, stair climbing, fall risk, or independence, the clinical value may be far less than the headlines suggest. In longevity medicine, the objective is not to build the largest MRI image. It is to preserve power, mobility, and resilience. So, where does Bimagrumab fit? Bimagrumab is not gene therapy, and I've talked to this in the previous episode. It is a monoclonal antibody. It blocks activin type 2 receptors, particularly the receptor known as ACT, RIIB. Myostatin and activins use these receptors to transmit signals that restrain muscle growth. By blocking that signal, Bimagrumab can increase lean mass. Yeah, it also produces reductions in fat mass in clinical research, including studies involving people with obesity and type 2 diabetes. This makes Bimagrumab highly interesting in the GLP-1 era and combination thereof. One of the central concerns during major weight loss is preservation of lean muscle tissue. A drug that reduces fat while preserving or even increasing muscle could theoretically change obesity treatment. It's the next step. But Bimagrumab also teaches us humility. The drug has repeatedly shown that increased lean mass is biologically achievable. Yet, translating that increase into consistent clinical, meaningful functioning improvement has been more difficult. Once again, mass is not strength, strength is not power. And none of those automatically guarantees us or equals longer life. Bimagrumab is nevertheless a valuable proof of concept. We're pretty far with it. I like it, but we still need more studies. It shows that the activin myostatin system can be manipulated pharmacologically without permanently, right, inserting gene instructions, which is good. That may offer a more controllable pathway than gene therapy, especially while the field is still learning which patients benefit. The biohacker argument usually sounds like this. Regulation moves too slow. Muscle is essential for longevity. Follistatin has a plausible mechanism, right? Early disease trials have not revealed catastrophic toxicity. Prominent individuals have already taken it. And somebody has to be first. There is truth inside parts of that argument. Regulatory systems can be slow. Medical innovation often begins with courageous participants accepting uncertainty. Yes. And many effective therapies initially look radical. But real scientific pioneers do not eliminate measurement, oversight, and transparency. Sometimes, COVID-19, they increase them. A legitimate early stage experiment needs a clearly defined product, independent manufacturing verification, sterility and purity testing. I mean, I can go through this, right? GMP facilities, you go on and on, accurate dosing, These things need to be established. We can't just registered protocols. We can't just jump on this thing. And I know a lot of you are going to want to jump on this thing. And if you're in the bodybuilding community or the fitness community, you're going all in on this because you've done it with peptides. Don't do it. This is another level. It's just another level, especially with the gene editing. So Yeah, we need the long-term follow-ups. We need adverse event reporting, and we need to publish the negative and positive results. Without those safeguards, being first can simply mean accepting risks that nobody is properly documented and that you're not ready for, right? This is not disruption. This is information loss. So, here you go. Ring the bell notification, invite friends, we keep going. I keep reviewing this. I'm going to tell you when things change. I'm going to bring it back up when we get results from all the trials going on. Myostatin signaling, here's an extra, interacts with cardiac biology, but animals and human finding cannot be reduced to blocking myostatin. Makes the heart stronger. An intervention intended to alter systemic growth signaling should include appropriate cardiovascular surveillance. More tissue is not automatically healthier tissue. So there's a lot to look at here. So this is the final verdict. Myostatin is real. Follistatin is real. The ability to manipulate this pathway is real. And the possibility that these technologies could help patients with devastating muscle disease is genuinely exciting. The biohacker story is not fabricated from nothing. It is built on legitimate biology, but it then leaps across an enormous evidence gap from this pathway regulates muscle to this unapproved formulation will safely increase muscle, reduce aging, and extend life in healthy adults. That conclusion has not been earned.